Dementia encompasses a range of neurodegenerative conditions that lead to memory loss and cognitive deficiencies and affect some 55 million people worldwide. Yet despite its prevalence, there are few effective treatments, in part because scientists still don't understand how exactly dementia arises on a cellular and molecular level. Now, a team led by scientists at Harvard Medical School and Harvard T.H. Chan School of Public Health has made progress in unraveling the mechanism underlying a type of dementia that strikes early in life. In a study published October 7 in Nature Communications, researchers discovered that a genetic form of frontotemporal dementia, FTD, is associated with accumulation of specific lipids in the brain, and this accumulation results from a protein deficiency that interferes with cell metabolism. The results, based on experiments in human brain cells and in animal models, provide new insights into FTD that could inform the design of new therapies. Additionally, the findings highlight a mechanism of metabolic disruption that may be relevant in other forms of neurodegeneration, the researchers said. A black box. There are several different types of dementia, each with complicated genetics that involve various mutations. FTD, characterized by a loss of cells in the frontal and temporal lobes of the brain, accounts for 5 to 10% of dementia cases. Often diagnosed in patients between 45 and 65 years old, the genetic forms tend to cluster in families. Around 15% of the time, FTD is linked to a specific mutation in the GRN gene, which causes brain cells to stop making a protein called progranulin. Previous studies have linked progranulin to parts of the cell called lysosomes, which are responsible for cleanup and other metabolic activities in cells. However, the function of the protein, including its role in the lysosome, has remained sort of a black box, said co-senior author Wade Harper, the Burt and Natalie Valley Professor of Molecular Pathology in the Department of Cell Biology in the Blavatnik Institute at HMS. Harper collaborated on the study with co-senior authors Tobias Walther and Robert Faries Jr., who were professors of cell biology at HMS and professors of molecular metabolism at Harvard Chan School when they conducted the research, as well as lead authors Sebastian Boland, a former research fellow. The researchers initially found that progranulin-deficient human cell lines in mouse brains, as well as brain cells from patients with FTD, had an accumulation of gangliosides, lipids commonly found throughout the nervous system. Next, the team used recently developed technology for purifying lysosomes to analyze the types and amounts of proteins and lipids present inside them. Using this technique, the scientists found that lysosomes in these cells and tissues from brains with FTD had reduced levels of progranulin, as well as lower than normal levels of a lipid called BMP, which is required to break down gangliosides, the lipids commonly found in the central nervous system. However, when researchers added BMP to cells, they observed that these cells accumulated far lower levels of gangliosides. Together, the findings suggest that progranulin in lysosomes helps maintain the BMP levels needed to prevent gangliosides from accumulating in brain cells, buildup that may contribute to FTD. Thanks for watching. Please subscribe my channel for more videos.